Welcome to the Fundamentals lecture for Chem 118, Experiment 3. This time we're going to be talking about a phenomenon known as regiospecificity. And we're going to do that by investigating a reaction known as electrophilic aromatic substitution. And then finally today we're going to talk a little bit about infrared spectroscopy, which will be our final analytical technique, which we'll be using for the rest of the semester to characterize some of the compounds that we create. So, without any more delay, let's get right to the question for the week. The question for the week is, when we use electrophilic aromatic substitution to nitrate bromobenzene, where and in what order do we expect these nitrations to take place? By the end of the week, we will have experimentally confirmed the location and the order of nitrations in this reaction. Electrophilic aromatic substitution reactions, as the name implies, gives us a way to substitute an electrophile for one of the hydrogens on the ring of an aromatic species, like this benzene ring. And the mechanism takes place because pi electrons, even when they're in an aromatic system, are still not held as strongly as sigma bonding electrons are. So they're weakly nucleophilic. And when we feed them an electrophile, something that's really, really electrophilic, they simply can't resist attacking that electron-hungry electrophile. When this takes place, we create what's known as a sigma complex, because we've traded a pi bond for a new sigma bond. So in this case, the first step is energetically driven by the fact that a pi bond is traded for a sigma bond. However, in the process of doing so, the ring loses its aromaticity. And this is not energetically favorable, so the ring naturally looks immediately for a way to recover the aromaticity. And the way that it does this is by losing a proton. The species here, the intermediate, is so very acidic that really anything, even remotely basic, is going to be basic enough to abstract the hydrogen, the proton, from that tetrahedral center, and in doing so, allow the ring to recover its aromaticity. This is the final step of electrophilic aromatic substitution. And as you can see, we've replaced what was originally a hydrogen on the ring of benzene with a new electrophile substrate. A more specific example of how this can take place is the reaction between benzene and nitric and sulfuric acids in combination. The reason for combining nitric and sulfuric acids is this. Sulfuric acid is a little bit stronger than nitric acid, and it's so strong, in fact, that just a small amount of nitric acid can actually become protonated in the presence of sulfuric acid. When this happens, the protonated nitric acid has the option to lose water as a leaving group. And when this water is lost as a leaving group, we create something called the nitronium ion, which is isoelectronic with carbon dioxide, but carries a positive charge on the central nitrogen. This is an extremely electrophilic species, and it will be attacked very rapidly by a benzene ring. Now, the extent to which this reaction takes place has to do with the electron density within the ring and the stability of the carbocation intermediate which is formed. And both of these factors are going to become very important when we start to consider what happens when this reaction takes place with substituted benzenes. When we perform this reaction in lab, you'll notice a very dark orange colored gas forming. This is oxides of nitrogen. It's actually a mixture of oxides of nitrogen. And it is also extremely toxic by inhalation. So while we are generating this inside the fume hood, naturally all of these standard rules will apply as far as keeping your sash at operating height and keeping yourself on the correct side of that sash. This is one of the more hazardous materials that we generate during this semester. So be absolutely sure that you avoid inhaling any of the nitrogen oxide gases. So let's begin thinking about the effect of substituents on the reactivity of rings in these kinds of reactions by considering first benzene. Now let's make a couple copies of benzene and make some small changes and see what happens. Let's exchange one of the substituents here for some other substituent, which is electron donating. That means that it's putting more electrons into the ring pi system. Well, this would increase electron density there and of course make it a better nucleophile and therefore a more reactive ring. Conversely, if we were to replace a ring hydrogen with some type of substituent which is electron withdrawing, which means it pulls the electrons out of the ring somehow, we expect to have a ring which is less reactive in electrophilic aromatic substitutions. So the first step in assessing how a substituted benzene will react is to consider whether or not the substituent donates or withdraws ring electrons. Let's use a specific example that we'll be looking at this week: nitrates. Nitrates are deactivating to rings because they can produce resonance contributors in which the ring electrons are outside of the ring itself. For example, we can draw this resonance contributor in which we've moved some of those ring pi electrons up onto the bond between the nitrogen and carbon. Not only that, but we can draw additional resonance contributors in which we still have the same condition. The aromaticity has been lost and some of the electron density has been pulled out of the ring. So in all of these resonance contributors, we can see that there is positive charge on the ring, meaning that the resonance hybrid itself is expected to be less reactive in an electrophilic aromatic substitution, because the effect of the nitrate is to withdraw electron density from the location that we need to be nucleophilic. But the presence of a nitrate on a benzene ring affects more than just the overall reactivity. It affects the reactivity at specific locations differently. I'm going to demonstrate this by considering what happens when we add an electrophile to the ortho position, which is the one adjacent to the substitution. Later we'll consider the meta and para positions as well. So let's create the sigma complex with the electrophile added at the ortho site. When we do this, you can see that I've drawn at least one resonance contributor in which I have the positive charge on a carbon directly adjacent to a positively charged nitrogen from the nitrate. I can go on to draw a few more of my resonance contributors and then construct a resonance hybrid and consider its stability. As you can see, we have a partial positive charge adjacent to a formal positive charge in the resonance hybrid for this particular intermediate. That means that when the electrophile is attempting to substitute at the ortho position, the transition state associated with that addition of the electrophile to form the sigma complex must also be higher in energy. Translation: this is a highly deactivated reaction when nitrate is present. However, when you consider what goes on when the electrophile is added at the meta position, you see that the positive charge is never located directly adjacent to the positively charged nitrogen. So this location is deactivated less. This effect can be shown graphically using reaction coordinate diagrams. Remember, we have intermediates that are sigma complexes at the ortho, meta, and para positions potentially. But we already discussed how ortho and para intermediates have a clash of positive charges up there where the nitrate is present. And of course that the meta does not suffer from this problem. So what this means about the reaction coordinate diagram is that for the ortho and the para, we expect to see fairly similar reaction coordinates. But for the meta pathway, because the intermediate's energy has been reduced, the Hammond postulate tells us the transition state energy must also be reduced. And because the formation of the sigma complex is the rate limiting step, we know that that means the reaction should run faster at the meta position. In this case, we expect that when we attempt to modify nitrobenzene through an electrophilic aromatic substitution reaction, that we will preferentially form the meta product. The other substituent that we'll be considering this week is bromine. Bromine is a member of the halogens, which is an unusual set of substituents in the sense that they deactivate rings overall, but that they have an ortho-para-directing effect. This stems from the fact that the electronegativity of bromine causes it to withdraw electrons inductively from the ring, but that it has available p orbitals which can allow it to participate in resonance in such a way that it stabilizes the transition states and intermediates for ortho and para reactions. So in the case of nitrates and bromines, we have two different types of directing effects. Nitrates direct meta, bromine directs ortho-para. IR spectroscopy, sometimes called FTIR spectroscopy, when using certain pieces of equipment, like the ones in our lab, is very similar to UV visible spectroscopy in the sense that we're simply measuring the absorbance of a very particular wavelength of light by a sample. Where IR spectroscopy often differs from UV visible spectroscopy, however, is that we tend to report in percent transmittance rather than percent absorbance. This is because the percent absorbance allows us to apply Beer's law in UV vis, but in the case of IR, we want to coax out smaller signals in the spectrum most of the time, and this is done better using the percent transmittance, which is directly measured by the instrument. We also use a different x-axis. We use something known as wave number, because we'd like to report on the frequency of light, but that gives us a very unwieldy number to deal with. And in the infrared region, wave numbers of lights reported in reciprocal centimeters gives us numbers that are more manageable, 500, 1,000, 2,000, etc. We've already discussed how ultraviolet visible spectroscopy is based upon the absorption of light in electronic transitions. In that case, we look mainly at the transitions of pi electrons. But the source of the absorption in infrared is something quite different. And to explain this, I'm going to start with the molecule in the top right hand of your screen here. This is a molecule called fluorine. That's fluorine with an E, not an I, so not to be confused with the element. Now, fluorine doesn't look like this. I've drawn a beautiful picture of it with all perfect idealized bond lengths and angles. But we would never expect to see a fluorine molecule that looks like this. The truth is that in nature, a fluorine molecule at any temperature but absolute zero behaves more like this. It's constantly moving, vibrating, and twisting, all of its bonds moving independently of one another to a certain extent. And it's these molecular vibrations that are the source of infrared light absorptions. So if you think of these vibrating bonds as simple weights and springs like in your physics course, remember that the heavier the weight and the stronger the spring, the higher the frequency is expected to be in a given vibration. This means that smaller pairs of atoms, for example carbon and hydrogen, are expected to have higher frequency vibrations. And furthermore, that the stronger the bond is between the two, the higher the frequency the vibration should be as well. If we have larger atoms, for example carbon and oxygen, we have a situation where we expect lower frequency bond vibrations. And just like joining the same two weights by a weaker spring should result in a lower frequency vibration, so does joining two atoms together of the same mass with a weaker bond. Case in point, a carbon-oxygen double bond should have a higher vibrational frequency than a carbon-oxygen single bond. This is the source of the x-axis of our spectrum. The selection rule for infrared spectroscopy is this. And anytime a dipole changes in response to a bond vibration, there's a potential for IR absorption. In other words, when we have a bond like, for example, a symmetrical alkene here that's just stretching perfectly straight, that there should be very little or no change in a dipole because there should be very little or no dipole present to begin with in the bond. But a bond of higher polarity, for example, these carbonyl bonds down here, when they stretch by just a small amount, can create a much larger difference in the dipole. And it's this change in dipole that is the selection rule for infrared spectroscopy. So the polarity of a bond has something to do with how strongly it's going to absorb infrared light of a particular frequency. And we can apply these to a basic correlation table for infrared spectroscopy that I've shown on the bottom right, in which we have situations where increasingly polar bonds absorb with increasing strength. Our CHs, CC, and CN bonds up near the top, and our OHs and CO bonds down near the bottom. Similarly, we have certain trends that we can follow along the x-axis. For example, we have much smaller pairs of bonded atoms that are absorbing at higher wave numbers, whereas we have these much larger bonded atoms which are absorbing in regions where we expect lower frequencies to take place. And this trend continues and follows the bond multiplicities as well. Knowing that a stronger bond should have a higher vibrational frequency, we see that a carbon-nitrogen triple bond has a higher frequency absorption than a carbon-nitrogen double bond, which is higher in turn than a carbon-nitrogen single bond. And similar trends exist for other pairs of atoms, such as carbon-oxygen and carbon-carbon. There's a correlation table of these spectra available in your lab manual that shows some approximate locations and intensities of peaks that you might encounter. We collect IR spectra using sodium chloride plates to support our samples. We do this because every other conventional material that we might use in a spectroscopy experiment actually absorbs infrared light fairly strongly. Now, the consequence of this is that we have to be very careful how we handle them, because as we all know, sodium chloride is very water soluble. So we can only use this technique using organic solvents and when we very carefully handle the plates so that we don't touch the surface of the plate with our fingers. Shown in this slide is a photograph of a properly prepared sodium chloride plate, in which a thin film of the material to be analyzed has been created by dropping a small amount of a solution of that material in volatile solvent, such as dichloromethane, has been placed on the surface of the plate, then allowed to evaporate. If this thin film is of the appropriate thickness, what will occur is that the beam source will generate a spectrum of infrared light, which will then encounter the disk, and a portion of that infrared spectrum will be absorbed. Of course, the remaining light should continue onward to a detector, and the instrument simply determines which wavelengths of light have been absorbed and which have not. So let's pause now to take a bit of a review on our fundamentals before we jump into the reaction for the week. We started by discussing electrophilic aromatic substitution. And we talked about how ring pi electrons attack an electrophile, generating what's known as a sigma complex. And that this sigma complex is then quickly deprotonated to reestablish the aromaticity. We talked about how substituted benzenes have distinct reactive sites, and that substituents can either increase or decrease the reactivity of the ring depending on whether they donate or remove electrons from the ring. We also talked about ortho, meta, and para positions, and how we can have specific activation and deactivation at different sites, which can be analyzed using the resonance hybrid and used to predict which type of product should form. And finally, we discussed a little bit about infrared spectroscopy and how bond vibration is the source of absorption in this technique. And then we talked about a few ways that we can find trends in the spectra and analyze and predict where bond vibration should absorb. We discussed how they're reported as percent transmittance and wave number, and how we use sodium chloride plates specifically as our sample supports because most other materials will absorb in that region. Now let's get back to this week's reaction. We're going to nitrate bromobenzene in an electrophilic aromatic substitution using nitric acid and sulfuric acid to form a nitronium electrophile. And the question we're asking ourselves here is, where does the first nitrate go? I've depicted it here in this example with a bond going directly to the center of the ring. And this is the organic chemist's code for I don't know where on the ring this is. Our job is to figure out exactly where that bond goes. Then we want to determine when a second nitration occurs. Where does that take place? And if a third nitration happens, where does that go? So we'll need to consider a few important questions here. First, how will we determine that we've nitrated the product at all? Second, how will we determine how many nitrations have taken place? And third, how will we determine the position of each nitration on the ring? So let's think about the techniques available to us in the lab and see if we can come up with an analytical strategy which will allow us to answer all of the necessary questions to complete our study. Here's a table in which I have 3 columns, each asking for a different thing. The first, did we nitrate the compound? The second, how many nitrations took place? And the third, where are they? Could UV visible spectroscopy tell us that we've nitrated bromobenzene? Well, the answer in this question is maybe. UV visible spectroscopy would be capable of determining that we've extended conjugation in the compound. We could do that simply by noting the pi to pi star transition absorption. But there's more than one way to extend conjugation in a molecule, and UV vis alone wouldn't be able to tell us that it was nitration that did the job. For the number of nitrations, we also would expect it to be fairly good. We could again, see that the pi-pi star transition has changed, but we couldn't really necessarily say that it's because we've nitrated bromobenzene, as other modifications would accomplish the same thing. And as far as the position of nitrations, whether it could tell us if we have 2 nitrations, whether it's para, ortho, or meta to one another, it's actually quite poor for that. In the absence of a reference spectrum, we'd have a very hard time using UV vis to determine this. So how about FTIR? Could it tell us that we've nitrated bromobenzene? Well, most definitely. FTIR is outstanding at detecting functional groups because functional groups tend to be where polar bonds are located. And polar bonds, as we know, create good, strong infrared absorbances. As for the number of nitrations, it's not going to be nearly as helpful, because we can't ever be sure if we only see what appears to be one nitrate if there aren't other nitrate signals just beneath it. Similarly, it's not going to help us with determining the position of nitrations. In the absence of a reference spectrum, FTIR would not be very helpful. How about GCMS? Could that tell us we've nitrated our product? It certainly could. It shouldn't be that difficult to run a GCMS and note that the mass of the molecular ion and masses of fragments correspond well to a compound which has been nitrated. Could it tell us the number of nitrations? Yes, it probably could. Again, because each time we add a nitrate group, we're adding a very specific mass to the compound, and we're creating bonds which fragment in known ways. How about the position of the nitrations? In this case, unfortunately, GCMS is not going to be terribly helpful. Once again, without any reference spectrum to go by, it's going to be tough to tell whether a dinitrated benzene product has the nitrates ortho, meta, or para to one another. Finally, let's think about proton NMR. Could that tell us that we've nitrated our product? Well, of course not, because there are no protons in a nitrate group. So proton NMR would only be good at telling us that we're missing protons. It won't tell us that we're missing them because of a nitrate group. It could be any other functional group that doesn't contain a hydrogen. However, if we combine proton NMR with another observation showing that we have nitrated and only nitrated our compound, could we tell the number of nitrations that took place? In this case, the answer is yes, because we can simply detect where protons are And assuming our FTIR data or GCMS data tell us that nitration has taken place, we can simply infer that anywhere we're missing a proton, we must now have a nitrate. And finally, could Proton NMR bail us out here and help us tell the position of nitrations? The answer again is yes, it could. And this is because we simply can locate all the hydrogens on the ring based on their couplings, and that will tell us the locations where we have removed or replaced those protons. And of course, our FTIR data tells us that those replacements could only be done using nitrate. So by combining at least two techniques, we can complete our argument. The point here being that there is no single magic bullet. There is no perfect technique which will always tell you everything you need to know. And this week is a great example of that. We're going to have to combine FTIR and Proton NMR analysis in order to complete our story. So we've decided to use IR spectroscopy to detect nitrate groups that have been added to our bromobenzene molecule. So what do we expect to see here? Well, there are two very important bond vibrations that nitrates introduce into molecules which make them so easy to detect by IR. Those are the symmetric stretch and the asymmetric stretch, shown here. These two different stretching modes have slightly different frequencies and therefore have slightly different absorptions in infrared spectroscopy. But because we're dealing with bonds vibrating here that have nearly complete charge separation, remember the nitrogen in a nitrate has a positive formal charge, and one of the oxygens at least has a negative. In truth, they both have about a half a negative. We expect to see a very, very strong absorbance. And in fact, that's exactly what we see, with the symmetric stretch having an absorbance right around 1350 wave numbers, and the asymmetric stretch somewhere just above 1500. This region of the IR spectrum is usually otherwise fairly empty. So it's very, very simple to detect the presence of at least one nitrate on your compound simply by looking for these two very pronounced absorbances. But remember, we've already decided that IR spectroscopy can't tell us where the nitrates went. It can only tell us that they're there. So in order to determine the location of those nitrates, we have to resort to our Proton NMR experiment. So here I've drawn three potential regioisomers of nitrobromobenzene. One in the ortho position, one in the meta position, and one in the para position. Now let's compare them and see if we can convince ourselves that their NMR spectra will be distinct from one another. Let's start by looking for the chemically distinct hydrogens. In the case of para, we see 2, whereas in the case of ortho and meta, we see 4 distinct hydrogens. So if the nitrate is located at the para position in a single nitration, it should have a very distinctive spectrum, containing just 2 doublets corresponding to the 2 different types of ring protons present. But what about the others? The other 2, the ortho and the meta, each have 4 chemically distinct hydrogens, so they should have 4 distinct resonances unless there's accidental degeneracy. So, can we tell them apart? Well, let's take a look at multiplicities. Notice the couplings here. We expect the green to be a doublet, the yellow to be a doublet of doublets, the orange to be a doublet, and the red to be a singlet in the case of our meta-substituted product. Now take a moment and try to figure out the multiplicities of our next one, the ortho-substituted. We get a little bit more complicated spectrum, don't we? This is because we've got more of the hydrogens adjacent to one another. So the orange and the red proton resonances will now be more complex. We'll have a doublet of doublets in the case of the orange proton, and a doublet in the case of the red proton. So the number of resonances and their multiplicities should give us a way to determine whether or not we have a nitrate at a specific location on our bromobenzene ring. And a word of warning here, there are also long-range couplings which can go through 4 bonds that add up to about 2 Hertz in aromatic systems. So you may see even more fine structure on top of these multiplets when you observe them in your NMR spectrum. So be aware of that. You might see them, but those are actually 4-bond couplings, not 3-bond couplings. Something for you to think about this week is which dinitrated products might form. There are actually 6 different permutations of dinitrated products, some of which we might predict and others of which we might not, based upon what we know about directing effects. Nonetheless, you should consider what the proton NMR spectrum of each of these would look like, and how you would prove exactly where a second nitrate went when it was attached to the ring. When you're done with that, you might even want to consider trinitrated products and see how many different isomers there are and whether or not you could distinguish them using NMR spectroscopy. Here's a spoiler alert. We won't be making any trinitrated bromobenzene products because the Navy uses that material as underwater explosive ordnance. So naturally, we're not going to give you a reaction that would produce that. Nonetheless, it's worth thinking about. If a trinitrated product formed, how would you distinguish it? This brings us to our goals for the week. Our goals in lab are the following. First, nitrate bromobenzene once or twice using an electrophilic aromatic substitution reaction. Next, we'll use IR spectroscopy to confirm that whichever product we formed has nitrate groups and only nitrate groups on it. Next, we'll use proton NMR to determine the position of those nitrations. And finally, we'll formulate an explanation for exactly why certain nitrates go into certain positions in a specific order when this reaction takes place. We'll see everybody in the lab for that next week, and we'll see you on Monday for our recitation, where we'll discuss how we're going to accomplish this reaction safely and effectively in the lab.